So we've got to get through the metabolism chapter today. Remember, I told you we were going to do a lot of picking and choosing with this chapter, so hopefully you didn't read the whole thing. Hopefully you have looked over cellular respiration a little bit to remind yourselves, because we're not going to have time to go over it as in-depth as you did in general biology. That's not the only form of metabolism that we're going to be talking about today. We're also going to look at what we do with things like proteins and fats. So, as we're looking at that metabolism, we're also going to think about where we get these nutrients from. Okay, so some of this is kind of repetitive because we've already talked a little bit about macromolecules as well, right? What were the 4 types of macromolecules? Lipids, proteins, carbs, and nucleic acids. Exactly. So you already know a thing or two about the nutrients. We'll just be adding a few more details today. So when we look at the things that we're supposed to be learning in this chapter, the most important things are going to be here in part 2. That's where we're going to spend the majority of our time. We're going to talk a little bit about different metabolic states, really just comparing like what kind of metabolism happens after we eat a meal versus when we go into starvation mode when it's been a while since we've eaten a meal. And we will also be looking a little bit at, what are the different types of nutrients as we go through this stuff. So if we're talking about metabolism, we have to think about what metabolism is. Again, what is metabolism? It's not always breaking down, right? We've talked about the breaking down type of metabolism. What's that? Catabolism. Good. What's the other type? Anabolism. Good. Where we're building things. So anytime we're talking about metabolism, we're literally talking about something related to chemical bonds. Either we're breaking them or we're building them up. That is what metabolism is in the body. And we have these 2 different types. We've talked a little bit about catabolism because we know that that happens in the digestive system, right? Because we're taking these huge macromolecules and we're breaking them down into their monomeric units. But where else would we see catabolism happening pretty regularly? What?
Turning glycogen into glucose.
Yeah.
Okay, so breaking down storage molecules into their smaller parts so that we can use those in what? Respiration, cellular respiration to make energy, ATP. Because remember, what's the first step in cellular respiration, at least if we're focusing on aerobic respiration? Start with the G. Glycolysis. Good. Glycolysis, which has lysis in the name, which means splitting, right? So we'll be talking about a fair amount of catabolism today. When do you think anabolism would be most important, like over a person's lifespan?
When you're a baby.
Yeah, during maturation into adulthood, for sure. When else? During repair, right? Obviously, the body is going to have to make more stuff. Also, today we're going to see that with nutrient processing, if we have an overabundance of a specific type of nutrient, we have different ways that we can build it into things to store it. So, keep those thoughts in the back of your mind as we talk about stuff today. We've already talked about the purpose of anabolism and catabolism. I want to give you a couple of other thoughts before we move into our nutrients and what we're going to do with them. Since a lot of what we're going to be talking about is how we make ATP, because that's a big point for bringing nutrients into the body, right? We keep talking about that in the digestive system. And then again here, let's remind ourselves, what is ATP? Well, I know it's energy, but what does it stand for? Adenosine triphosphate. Very good. What does tri mean? 3. So we have 3 phosphate groups stuck onto this adenosine, which you don't have to know the chemical formula for. Thank goodness. When we make ATP, what we're going to be doing is we're going to be taking ADP and adding a phosphate group to it. What does the D stand for? Di, exactly. So ADP is just the adenosine with 2 phosphate groups. So if we're going to turn that into ATP, we just need to stick an extra phosphate onto it. Whenever we add a phosphate group to something, we call that phosphorylation. So when we study cellular respiration, we're going to see that there are 2 different ways that we can stick this phosphate group onto ADP. Through substrate level or oxidative phosphorylation. And I'm going to tell you right now, we're going to see way more substrate level, at least as far as where it happens and when it happens. What is a substrate? Because you probably learned about those all the way back in general biology, maybe when you studied enzymes. Nobody? Are you guys still waking up because it's so rainy outside? That's what the enzyme can bind to. That's exactly right. And that's what I'm showing you in this picture here. The big purple thing is the enzyme, and they have the substrate labeled for us. So notice our substrate here has a phosphate group, and our enzyme is holding that substrate close to ADP so that ADP can steal the phosphate group and turn into ATP. So with substrate level phosphorylation, we see the phosphate group getting pulled off of something else so that we can add it to ADP. With oxidative phosphorylation, we're going to be working with inorganic phosphate, which don't worry about every single word on this slide, but notice our inorganic phosphate right here, it's not bound to anything else, and so they often abbreviate that as PI for inorganic phosphate. And we're going to be adding that to ADP to make ATP during a specific point in cellular respiration, right at the very end of it. What process happens at the very end? What was that major phase? The ETC, the electron transport chain, good. And at the very end of that, we have something called chemiosmosis happening, which is going to help us out with this ATP production. So just keep that in mind. We're only going to really be seeing the oxidative phosphorylation happening at the very end of cellular respiration. And that involves inorganic phosphates. So just these phosphates that are free floating around by themselves, we're not having to steal them from anybody else to add them to ADP. All right, well, before we can talk about cellular respiration, it's always a good idea to remind ourselves about the primary fuel source for that, carbohydrates. You guys already know a thing or two about carbs. What were the smallest ones? Monosaccharides. Very good, because mono means what? One. If we add 2 monosaccharides together, what do we make? Disaccharides, exactly. And if we have a bunch of monosaccharides stuck together, what have we got? A polysaccharide. What was the most important one? Starch. We're going to be seeing starch again. Good. What was an important monosaccharide? Glucose. Obviously, we're going to be talking about that a fair amount today, so we can't forget about it. It's not the only one you need to know, though, right? So I've got those reminders for you as we go through these slides. So notice starch, one of our more complex carbohydrates, comes from anything plant-based, basically. So anything that we eat that was made from grains, like cereal or bread or granola bars. Also, a lot of your fresh veggies that you eat are going to have a fair amount of starch. Another type of complex carbohydrate that I want you to be aware of is fiber, because it is something that we hear a lot about in our diets. And what I want you to know is that there are different forms of fiber. Some are soluble, meaning that they're fully dissolved in water. Others are not soluble and add to a more solid component in our food stuff. So on the slide, I've got pectin for you, which is actually thought to help lower blood cholesterol levels. And we find it in things like citrus fruits and apple peels. To give you an example of an insoluble fiber, we have cellulose. And didn't we talk about cellulose before? Yeah, when did we talk about cellulose? With the bacteria, exactly, because the bacteria that live where? In the lumen of the large intestine. Good. Those bacteria could metabolize it. We couldn't, right? Because we don't have the enzymes to break down cellulose. And notice on this slide I'm reminding you where cellulose comes from. Where does it come from? Plant cell walls, exactly. So anytime you eat something plant-based, you're going to be intaking cellulose. And even though we can't break it down, we know the bacteria metabolize it, and that helps with the production of vitamins that we need, like B and K, right? We also know, well, what I want you to know now, anyway, is that this solid component in the food stuff is important because it gives the intestines something to really push on when they're doing those peristaltic contractions to force the food stuff toward the rectum and the anus. If it was just a liquid, it would be a lot harder to move it. It's easier to move it if we have some stuff that isn't dissolved and is more of a solid. So even though I told you that the chyme was mostly a fluid, it's not totally a fluid. It's a little more like the consistency of like the mucus that we've talked about, where it's a bit thicker because of the cellulose, this insoluble fiber. Reminders of other carbohydrates. What was sucrose classified as? A disaccharide. Good. And we said that came from things like table sugar. So anything with added sugars is going to have sucrose. Lactose has that LACT in it because it comes from milk-based stuff, not just milk, but cheese and yogurt, anything that was made with milk products. We've also talked about maltose, another disaccharide, which we know comes from grains, or anything that was made with grains, like bread, cereal, beer, those kinds of things. Fructose and glucose come from a variety of substances, like fruits. That's why fruct is in the name for fructose. It comes from fruits. Honey is another good place where we find both of these sugars. And galactose has that LACT because it also comes from milk-based substances. So these are these disaccharides? They're all monosaccharides, right? Because we see glucose in there, as well as two others that we've discussed. Okay. So when we think about these carbohydrates, we know that glucose levels in the blood are going to increase anytime we eat a meal. Right? Because we've discussed that already, like when we talked about insulin and glucagon before. I'm giving you this figure. It doesn't come from your book. You don't have to memorize everything on it, but I'm highlighting some things. So notice they're showing us that right now we have a high concentration of glucose in the blood. And a lot of that glucose is being pulled out and used by a lot of the body tissues for cellular respiration to make ATP. Right now, after we've eaten a meal, though, we probably don't need to use all of that glucose for ATP production. So now's our chance to store it, and we can see two areas that store it. We have skeletal muscle and we've got the liver. And we can see that they both have the ability to store glucose as glycogen, which we've discussed before. Remember, that's the one that I always draw like a bunch of glucose blocks that are stuck together to help you remember that it is the glucose storage molecule. Notice that in the liver, there's another way that we can process our sugars. If the sugars are really high, what's the liver going to also make? Lipids, fats, exactly. We know triglyceride is an important lipid in the body. So notice that the liver is also converting glucose to triglycerides, which we can then store in adipose tissue. So yes, we are going to primarily focus on cellular respiration, but we have to keep in mind that there are other processes that can happen so that we can store glucose. And we're going to look at those a little bit more after we talk about cellular respiration. Any questions so far? No. All right. All right, then remind me, what are the major processes that are going to help us with cellular respiration? We already talked about the first one, guys. What was the first one? Glycolysis. Okay. And we know lysis always means splitting. What does glyco mean? Sugars. Sugars. So literally, we're going to see sugars being split into two during glycolysis, and we're going to focus on glucose here. What's another major process that's going to help us with cellular respiration? The Krebs cycle, or the citric acid cycle. And then what's the last one? The electron transport chain, which is coupled to chemiosmosis. And I have that 2 up there because there's going to be a transitional step or a transitional phase that helps us get what we make in glycolysis into the Krebs cycle. So we can't forget about that. Before I go on to the next part, if I were you, I would begin to make myself a chart. On your chart, you want all four of these phases of cellular respiration at the top. And then in your rows, you want things like where it happens, what it uses, and what it makes, because those are going to be the important things that I'm going to be emphasizing in this next bit. So go ahead, get a sheet of paper, make that table. It's going to save you a lot of time later. While you guys are finishing up those tables, why don't we go ahead and think about where these processes are going to be happening. What was the organelle within the cell that helps us with cellular respiration?
Mitochondria.
Mitochondria, very good. And anytime your books draw the mitochondria, they're kind of an egg shape, and then they've got like this squiggly line inside of them, right? So what that's showing us is that there's an outer mitochondrial membrane and an inner mitochondrial membrane. And that's important because that creates 2 different compartments within the mitochondria. The innermost compartment is called the matrix. And then we have this intermembrane space that's happening between our two membranes. Some books will call that an intramembrane space, saying it's within the two membranes. Outside of the mitochondria, what do we call the fluid that we're finding inside the cell? Interstitial is between cells out in body tissues, right? The cell is filled with cytoplasm, that's right. And sometimes you'll see that referred to as cytosol. So let's make a note about that as well. Our first step, glycolysis. Do you know where that's going to happen? Outside the mitochondria, very good. Out here in the cytoplasm. So that's where glycolysis is going to happen. And then what we make in glycolysis has to be converted to something else as we bring stuff inside the mitochondria. So that transitional step kind of happens both in the cytoplasm and within the mitochondria as we're converting what's made in glycolysis to something else. We're going to see what that is in a minute. Where does the Krebs cycle happen? In the matrix, on the very middle of the mitochondria, in the matrix. That's where the Krebs cycle happens. And then where's the electron transport chain located? The electron transport chain, remember, was made up of a bunch of proteins that were stuck in a membrane. Which membrane are they stuck in? Oh, I'm hearing guesses. Okay, let's remind ourselves. The ETC is located on the inner membrane. So when we're studying that fourth step, we're going to see substances moving from the matrix to the intermembrane space, and then from the intermembrane space back to the matrix. So we see a lot of movement across that inner membrane as we study the electron transport chain. One other thing that we need to keep in mind before we go any further. When we think about what we're using and what we're making here, we've got a chemical equation for that, which you probably had to learn in general biology. It started with something C6 something. All right, we had C6H12O6. What is that? That is glucose. Good. And then what else is used? O2, oxygen. Yeah, because it's aerobic respiration if we're in the mitochondria, right? So it has to use some oxygen. Does anybody remember how much? 6. Very good. And then that's going to make, obviously, ATP. But as we're making the ATP, we're changing this other stuff into other things. What were those other things? Carbon dioxide. How much carbon dioxide? 6 carbon dioxides. Good. And how many waters? 6. So basically you're adding 6 in front of everything because you have to balance your atoms on either side of the arrow. If we have 6 carbons over here in glucose, we have to track all 6 of those carbons. Similar deal with the hydrogens. If we have 12 over here, we have to make sure we have 12 on the other side. And we know that in addition to carbon dioxide and water, we're also obviously going to be making lots of ATP as well as some heat. And we'll talk about why the heat while we're talking through the steps. So kind of keep this in mind because it's going to help you with seeing some of the things that you obviously need to track in that table that we're making. So let's start with glycolysis. It is a multi-step process. You do not have to know every single step. But we break it down into 3 major phases within glycolysis. So remember, this is sugar splitting. At some point, we should see a sugar being split into 2 pieces. Before we can get there, though, we have to make our sugar less stable and more ready to react so that it will split. We're going to do that by investing some energy in this sugar activation step. Sometimes your books will call that an energy investment step. Either way. So notice what we're doing. We're taking a phosphate group from ATP and we're adding it to either end of glucose to make a different molecule. You don't have to know that name, that fructose 1,6-bisphosphate. You don't have to know that. That's an intermediate. But in your table, you want to make a note that one of the things that we're starting out with is glucose. So that's one of the things we're using. And another thing that we're using is 2 ATPs because we're doing that right here in the energy investment step, right? Or the sugar activation step. And the whole purpose of this is to make our sugar less stable.
So what it makes is basically the sugar less stable.
Exactly. Exactly. That's the whole point of adding this energy because it's kind of weird, right? We're using energy to make energy. Why would we do that? We're having to do that because that's the only way we're going to be able to split our sugar into 2, which is what we see in the next major phase, the sugar cleavage phase. Okay, so in this phase we can see our sugar being literally split in half. Once again, you don't need to know the names for the things that it's split into, but you do need to know that in the sugar cleavage phase, which comes after energy investment or sugar activation, we split. And this is an important thing to focus on because from here out, everything that we make per glucose should be an even number. Because we only had the little black dots there are representing the carbons. So we have 6 carbons up here still, right? Like the original 6 from our glucose. And then we're splitting that into 2 molecules that have 3 carbons. So if we're going to track all of these carbons, we're going to have to follow everything twice from here on out. In the last step, we have sugar oxidation and ATP formation. So this is when we actually get our energy production. That's not the only thing we're going to make, though. So in the what it makes part of your table, it's showing us that 4 ATPs are made, but is that net ATP production? Is that what we're actually getting out of glycolysis? No. How many net?
2.
Only 2 net. So you want to make a note about that in your table. It's only 2 because we used 2 already. So 2 out of the 4 are replacing the ones that we used initially.
So the end result, what it makes is 2 net ATPs.
That's right. That's right. Notice it's also showing us that 2 pyruvic acids are made. Sometimes your books will call that pyruvate. It's essentially the same thing.
So we add that in what it makes.
That would be another thing in the what it makes column, yep. And what else is made? NADH is how many? All right, so we've got to add that to our table as well. And you probably want to remind yourself what NADH is. Does anybody remember? I know it's been a while since you all have had to think about this stuff. It's an electron carrier. So NADH really isn't so important right now, but it will be important later when we get to the electron transport chain, because it's going to be one of the things that actually adds an electron to those proteins in the electron transport chain. So the values that I have in the boxes, is that how many are made per glucose?
No.
Yes. For every 1 glucose, we make all of that stuff. Notice the pyruvic acid has 3 carbons, right? So if we're making 2 of them, we're following all 6 of those carbons from that original glucose. They're also showing us at the bottom that glycolysis happens whether we have oxygen or not. If oxygen is not present, then we go to anaerobic respiration, where we convert that pyruvic acid to waste products like lactic acid. However, here we're focusing on true full cellular respiration, so aerobic respiration. We need to see what's going to happen to that pyruvic acid in the presence of oxygen. You guys with me so far? Any questions about what should be in your table?
Yeah, should we add also the pyruvic acid?
To the what it makes column?
Yeah.
Yes. So that's an important part of what we make at the end of glycolysis, because it's like we turned the glucose into these 2 pyruvates. And now the 2 pyruvates are going to be used in that transitional step. So you could just draw an arrow for your pyruvates going over to the transitional step in the what it uses part. So notice, just like I told you, I know this chart is showing us a lot, but focus on the red box first, because everything in the red box is in that transitional step. So notice we have our pyruvic acid, or pyruvates, from glycolysis that are going to be fed through this transitional phase. And well, as we do that, notice what we're making. We're making some carbon dioxide, or we're making more NADH, and we're making acetyl-CoA, which is important because that's what we're converting the pyruvic acid to so that we can get it into the Krebs cycle. So, that's just showing me 1 carbon dioxide, 1 NADH, and 1 acetyl-CoA. Are those the values per glucose? Can't be, because it's not even, right? We have 2 pyruvates, 2 pyruvic acids. So that transitional step has to happen twice for every glucose molecule, right? So you've got to pay attention to that. If I ask you how much is made per step, that is different than how much is made per glucose. Because everything after glycolysis has to happen twice per glucose. Because we turn glucose into 2 molecules, 2 pyruvic acids. So I would be making a note about that in my chart as well. So in the transitional step, you know what it uses, the pyruvic acid. You know what it makes. First time that we've seen, carbon dioxide. We also see NADH and acetyl-CoA, but make a note, after you add those 1s, that you would multiply those by 2 if I asked you per glucose. Okay, so once we create acetyl-CoA, now we're ready to get into the Krebs cycle.
1 second.
Yeah.
What it uses, we said?
Somebody help me out. What does the transitional step use? Just what it uses. What it uses, not what it makes, is just the pyruvic acid, right? Pyruvic acid. Well, it would. If I ask you about 1 transitional step, it can only do 1 at a time. But if I ask you per glucose, that step has to happen twice, right? Good questions this morning, guys.
Where's the transitional step happening?
It kind of happens between the cytoplasm and the mitochondrial matrix, mostly in the mitochondrial matrix.
In the matrix or in the membrane?
Let's just say within the mitochondria.
Okay.
All right, let's just keep it general. Because it happens as we move the pyruvic acid from the cytoplasm inside the mitochondria. So just know that's important for helping to get our carbons in to the Krebs cycle. Yeah? For the Krebs cycle, but they were still asking questions about the transitional step. Yeah. So for the Krebs cycle now, the thing that we made that still has our carbons, the acetyl-CoA, is one of the things that we use.
But if you say per glucose, then it will use 2.
For per glucose, this would have to happen twice. That's exactly right. So at the bottom of that part of your chart, you probably want to say something about multiply everything by 2 per glucose once again. And when we see what this makes, this is making a ton of stuff. How much carbon dioxide are we making per Krebs cycle? We only have 2 carbon dioxides being produced. That's exactly right. What else are we making? We're making NADH. How much?
3.
We're making 3 NADHs. What else are we making? FADH2. So we're seeing another electron carrier being made. And it's only making one of those. That's right. So there's one FADH2. So notice at the very bottom, it's also showing us that we get some ATP out of the Krebs cycle as well. 1 ATP every time we run the Krebs cycle. And it's also showing us that the phosphate that we're adding to the ADP to make the ATP is coming from another molecule. You don't need to know what GTP stands for. Not right now. In your upper level classes, you may have to. But just know that we're pulling a phosphate group off of that, because what do you want to bet? That TP, what do you think that probably stands for? If it's going from a TP to a DP, we're seeing a triphosphate being turned into a diphosphate, right? Which is telling us that we pulled a phosphate group off of there. So what kind of phosphorylation are we using? Not the oxidate, the substrate level. Very good. Just like that's also true for glycolysis. So I would make a note about that in the glycolysis column as well. That we saw substrate level phosphorylation there because we were pulling the phosphate groups back off of those sugars.
And that's also with GTP and glycolysis.
No. Remember, you don't have to know all of the intermediates. We're going over some of the highlights because we only have not even a full lecture to talk about this stuff today. Now, if I were to ask you how much is made per glucose, you know you'd multiply by 2, right? You've already put that in your column. So how much ATP would be made by the Krebs cycle per glucose? 2, that's right. And 6 NADH and 2 FADH2 and 4 carbon dioxides, right? Because we'd multiply all those things by 2 per glucose. All right, so from the Krebs cycle, we've got to follow our electron carriers over to the electron transport chain. So first off, notice what they're showing us. We can see the outer mitochondrial membrane up here. The inner one is down here. We can see the intermembrane space here, as well as the matrix down here. And just like I told you, we see some big purple proteins stuck in that inner membrane, right? That's what these big purple things are here. Those are the proteins for the electron transport chain.
Is it H plus or H minus?
It's H plus. So before we focus on those hydrogen ions, notice we've got our electron carriers that are coming up to the proteins, and they're donating electrons, those are E minuses, to the proteins. But are they doing it at the same point? No, they aren't. Who donates earlier? NADH does, because it's giving its electron to the very first protein in the chain. And that's important because that means that NADH will help us to make more ATP than FADH2 will.
Is that what we use?
Well, we use both, but the NADH will give us more ATP. That's why we make more NADH than we do FADH2. So electrons have a lot of energy. And as the proteins pass the electrons off from one to the next protein, they're doing other things besides just interacting with the electrons. They're pumping hydrogen ions into that intermembrane space. And that's important because what that's doing is it's establishing a gradient. We have a much higher concentration of hydrogen ions in the intermembrane space than we do in the matrix because we are actively pumping the hydrogens in a direction they don't want to go. Notice by the time we get to the last protein here that's going to interact with the electron, well, it's not going to just release that electron out to the fluids of the cell. We've got to do something with that high energy electron or it might cause problems for us. So notice what we're doing to it. We're handing it off to oxygen so that we can make water. So in your column for the electron transport chain, we're using the FADH2. We're using the NADH, and we're also using those 6 oxygens that we talked about in that equation at the start of all this. In the what it makes part, we know it's got to make 6 waters if we're using those 6 oxygens, right? Now, that was a lot of work, but there's a purpose for it. We're establishing this gradient all because of this last protein that we find at the very end, the part that's going to do the chemiosmosis for us. So that protein is ATP synthase. We know if it ends in ASE, it's got to be what? An enzyme. So this is an enzyme that synthesizes or makes ATP. And the way it's going to do that is by using mechanical energy to help us make this chemical energy ATP. And what do I mean by that? Well, notice what's happening. We have a hydrogen ion from the intermembrane space, and we can see that that hydrogen ion is passively moving along its gradient into the matrix through this protein. As the hydrogen ion moves through the protein, though, it causes this upper part to spin. Well, that's physical movement. That's mechanical energy that's happening right there. And the protein can then use that mechanical energy to help phosphorylate ADP to make our ATP. Notice that the ADP that we're phosphorylating, we're using an inorganic phosphate to do that. So what kind of phosphorylation are we seeing here? Oxidative, not substrate level. It's the only place. And technically, this makes most of our ATP.
So it's more than 2 ATP.
Oh, yeah.
Like, we don't multiply.
Like 16, right?
It's like, it all depends on exactly what cell you're in, because different cells have different enzymes. But for simplicity's sake, this process makes about 30 ATPs in total for all of cellular respiration. So this, somewhere around 28 or so.
That's for glucose we multiply by 2.
Not for this. For this, it's a little more complicated because we're not tracking those carbons anymore. We're following the NADH and FADH2 here. So you don't have to worry about that whole multiplying by 2 thing for this step.
I have a question. So in the column, what it uses, should we add also the ADP?
You can, because technically it is. Yeah,
Okay.
technically it is. But that would be true anytime we make ATP, right?
Right.
So as long as you know how ATP is made,
Okay.
you're okay.
So inorganic phosphate just means it's not attached to anything. It's just
Exactly.
free floating.
Exactly.
Why is it called inorganic then?
Because it's not attached to any carbons. So, carbons are found in organic molecules made by the body. So,
How many ATPs do we make here?
about 28. So, I have a summary figure. And notice it's telling you about for some of these values as well, because it really does vary. It depends on exactly what type of cell you're in, because different cells have different enzymes. I know it's showing us a lot, but one of the nice things about this figure is it's showing us where our steps are happening. It's also helping us to track our electron carriers, as well as our molecules that contain the carbons that we got from the glucose. It's showing us how much ATP is made in each step. And you don't have to memorize the values over here for the NADH and the FADH2, but do notice that it's just like I said, for every 1 NADH, we're making more ATP than we are for every 1 FADH2. About 1 more ATP for every NADH than FADH2. And they've got a summary for overall ATP production for aerobic respiration. So this is just another way to summarize it. I personally like the table as well because I think it makes it very concise and easy. Do you guys have any questions about cellular respiration before we move on?
What it happens, the chemiosmosis is in the intermembrane space, right?
Not exactly, because it's chemiosmosis is happening with one of those proteins that's embedded in the inner membrane. So really, that's another area where we're seeing stuff moving between the intermembrane space and the matrix, right?
Mm-hmm.
So it's happening within the mitochondria between those two spaces.
Okay.
Yep.
So if you're pumping out the H+,
Mm-hmm.
I think going like against the gradient would take energy to do that?
Yes, and the energy is coming from the electron. ATP isn't the only kind of energy. So even though that's the type of energy that we focus on the most, electrons are very high in energy. And that's why we don't want that just free floating around, because if it's high energy and it interacts with something else, it's going to alter whatever that something else is. That's why we need to contain the electrons and add them to oxygen, so oxygen will interact with water, with hydrogen to make water.
So that's what
It does that
Mhm.
whenever it reaches a high level of energy, right? So like
No, the
energy
electron
goes up.
has high energy. The electron has high energy. So we don't just, at the end of the, so think about what's happening here, guys. We have proteins that are essentially passing off this electron like a football. All right, and every time they have the electron, they're able to pump hydrogen ions into that intramembrane space, right, between our two membranes. It's a pump because it's using the energy from the electron. Notice this first one is going to pass off the electron until we get to another pump that can pump more hydrogen ions into the intramembrane space. And then we see that happening one last time over here. But this last protein, well, it can't just let the electron go. That could cause problems, and I don't want to get into what those problems are because that's a higher level concept. So notice what we're doing with the electron, though. We're adding it to oxygen so it will react with hydrogen and make water. That way we're containing those electrons. We're not allowing them to just float around inside the cell and cause problems.
So that electron belongs to the hydrogen initially also, right?
Yes. So are we good on the ETC?
Yes.
That clears some things up. All right. We obviously know when glucose levels in the body are high, we do cellular respiration. That's what we were just talking about. But remember, before we talked about what the carbs were, we also said that when glucose was high, we could store it as other things. So on this chart, I'm showing you one of the things that we can store it as, glycogen. Notice what they're showing us here. These little green blocks are our glucose molecules. And with the purple arrows, we can see that we're using energy to turn that into glycogen, which they're showing essentially as a bunch of little glucose blocks stuck together, kind of like the way I draw it, just a little bit prettier. You do need to know that that process has a name, glycogenesis. If we break that up, it's got genesis in the name, right? So what does genesis mean? To create or to form. It also has glycogen in the name. So it's literally telling us that with glycogenesis, we're making glycogen.
So the reason we're using the energy here is because we have extra energy as well.
Right. There's plenty of glucose right now in the blood. Well, and it's also really what it's showing us is anytime we make something in the body that needs energy, it requires energy to make that happen. And remember why we're storing this glycogen. Because eventually, if it's been a while since we've eaten, like if we skip lunch today, those glucose levels in the blood are going to drop. And that can be a real problem for what part of the body? The brain. Because remember, the brain is like a glucose vampire. The only fuel it will take is glucose. Other tissues will take other things. The heart will even take wastes like lactic acid and use it as fuel in cellular respiration. Not the brain, it's picky. So, you also do need to know that when we split up glycogen to make glucose, that's got a name too, glycogenolysis. So once again, we're seeing glycogen in the name, but now we're pairing that up with splitting, right? So that literally means glycogen splitting.
So that
Well, not yet. We have to take those glucose molecules, feed them into cells that will use them for cellular respiration, right? And it's a little more complicated than this. We're going to see that in a minute. Oh. If the glucose levels get really low, there's another process that the body can use, and this is a term that we've seen before, gluconeogenesis. What does that neo mean?
New.
It means new. So why are we forming new sugars? Yes, it's because the glucose levels are low in the blood, but like we didn't call glycogenesis. That was making sugars, right? And there was a glycogenolysis, which formed more glucose, but that was different from gluconeogenesis, where it was new sugar. Why is this new sugar?
It didn't enter the body yet.
Because we're using other molecules to make sugars, right? Notice what's happening in the skeletal muscle. We're taking a protein, we're breaking it down to its monomeric units, the amino acids, and then in the liver, we can convert those to glucose. Notice there's a similar thing happening with fats. Our triglycerides are being broken down, and specifically the glycerol group from triglyceride can be converted to glucose here in the liver. Remember, when we talked about the liver, yes, we said it makes bile, but we also said that an important function was processing of nutrients. So you're specifically seeing that happening here. The glucose levels dropped, and now it's the liver's job to get those levels back up by turning other things into glucose. And it's even showing us why that's so important. Because we need that glucose to help the brain make ATP. We're going to come back to this figure in a minute. We're going to look at other parts of it. But do you understand the part about the gluconeogenesis for now? All right, then let's move right along to our lipids. What are lipids again? Fats. They're fats. And so what was the most important lipid that we talked about? Triglyceride, which we just saw in that previous chart, right? Why does it have tri in the name? Because it has 3 fatty acids that hang on to that glycerol group. Very good. Here, I'm telling you a little more detail. Triglycerides come in 2 different flavors, saturated and unsaturated. So when you're reading those nutritional labels and they're talking about saturated fats or unsaturated fats, they're really talking about triglycerides. Obviously, we know animal-based products contain the bad fats, the saturated fats, right? So whether it's animal meat or something else that came from an animal, like cheese from its milk or butter, those kinds of things are going to contain higher levels of saturated fats, whereas plant-based things are going to contain the healthy fats, the unsaturated fats, and that's why I have things like vegetable oil and nuts on the screen here. Now, triglyceride is a very important complex lipid, but it's not the only one, so I also do want you to make a note about cholesterol. Once again, potentially harmful fat. Look at where it's coming from, animal-based things, as well as egg yolks and seafood. All right, so if we eat a meal that's high in lipid, what can we do with that lipid? Well, a lot of the same things that we saw with the glucose. Remember, glucose we could feed into cellular respiration to make ATP. Well, lipids are fuels as well. So notice if we eat a meal that causes the triglyceride levels to increase in the body, Well, now we can break up that triglyceride and we can feed its parts into different parts of cellular respiration. You don't have to know at exactly which small step within the process, but I do want you to know that glycerol gets added into glycolysis, whereas the fatty acids are going to be converted so that they can be turned into acetyl-CoA and added into the Krebs cycle. This chart shows us a little more detail with that. So we're going to focus on the blue arrows for right now. Notice it's still showing us triglycerides being broken down into their parts, and then those parts are being added just where we said they would. But it's giving that a name. So when we break down lipids, that's lipolysis, literally splitting lipids. And I have another fancy term in blue, ketogenesis. This is something that happens when we increase fat metabolism in the body. Like if someone is dieting and they're doing a low-carb diet. Or if somebody is in a fasting mode, they haven't eaten for a while, the body starts to break down its fat stores. And when we go over to more fat metabolism than sugar metabolism, we have some different wastes that build up in the body, these ketone bodies. The process of making them is ketogenesis, and the reason why we care about that is because they are acidic. And if they're acidic, what can they do to the blood potentially?
It's going to be more acidic.
They can change the pH, they can make it more acidic. What was that condition called? Acidosis. So far, we've only talked about respiratory acidosis, but in a minute, we're going to see this can create another type of acidosis.
So Atkins diet is the worst.
Well, you have to be careful with that, and that's not the only thing that can cause these ketone bodies to build up, but it is one of the things. Notice that if we have plenty of nutrients in the body, like after we've eaten a meal, when we're building up fat so that we can store them in adipose tissue, that's what the purple arrows are showing us, and we've got a term for that process as well, lipogenesis. Once again, genesis meaning formation, except in this case we're forming lipids, building them up to make triglyceride, which we store in adipose tissue. So because we can turn, things like carbs and things like lipids into some of the same molecules, that's how we can convert from one to another. In the liver, anyway, because the liver has the proper enzymes for that. One quick note about proteins. We need to see where they come from. We need to know a little bit about how we build them up and how we break them down in the body. Clearly, we get a lot of protein anytime we eat something from animals. These are considered complete proteins because they contain all of the essential amino acids that our bodies cannot make, that we have to ingest in order to make all of the proteins in our bodies. A protein is considered incomplete if it does not contain all of the essential amino acids. So things that come from plants won't contain complete proteins. They're incomplete. Which means that if someone is a vegetarian or a vegan, they have to be much more careful about having a more varied diet if they're going to get all of the amino acids that their bodies need. They can't just eat grain-based things. They have to do a mix of grains and things like legumes or beans. And that's what I'm showing you in this figure here. And I know this is saying 8 essential amino acids, whereas the last one was saying, or this one's saying 10, whereas the last one was saying 8. The reason why this one says 10 is because infants have a couple of extras that they cannot make. In an adult, there are 8 essential amino acids. You don't have to memorize exactly which ones are essential. I won't make you do that here. If you take a nutrition class, they'll probably make you. But notice what it is showing us for our vegetarians. If they're going to get all 8, they have to do a mix of things to get all of those because they're intaking incomplete proteins. All right, a couple of other things here. When it comes to making proteins in the body, we follow an all or none rule. If we do not have all of the amino acids that we need to make the protein, we don't even start the process. It would be a waste of energy. Also, if we're not intaking enough calories, we're going to end up starting to do more protein breakdown than synthesis or building up. So if a person is fasting, if it's been a while since they've eaten, one of the things that the body will start to do is it will start to break down its own proteins so that we can use them to bring those glucose levels back up for the brain. There's also something called nitrogen balance. And what this essentially tells us is it gives us a term for the amount of protein synthesis or building up versus the amount of protein lysis or breaking down. We say that there's a positive nitrogen balance in the body if we have more synthesis going on. There's a negative balance if we have more breaking down going on. And notice when those things happen, we have a positive balance during maturation, during tissue repair, when we have more anabolism going on, when the body's building up more things like more cells. It's negative when detrimental things are happening to the body, like infections or injuries or whatnot. You do not have to memorize this whole slide. But notice the yellow boxes contain amino acids. And notice that we can convert those amino acids to different things. So when it comes to breaking down proteins, depending upon the amino acids in that protein, we could add them to glycolysis to make pyruvic acid. We could add them at the end of that transitional step as acetyl-CoA. Or we could add them directly into the Krebs cycle. So we could add them to any of those first part, anything before the electron transport chain. So, since we know that proteins are going to be broken down more during starvation, I want us to look more at what happens with starvation here. But this is going to help us to summarize some other things that we talked about. So we're going back to that ugly figure that I showed you earlier that looks like it has so much going on. But I want you to notice that it has a lot of processes that we already discussed. So for one thing, we're seeing triglycerides from the adipose tissue being broken down. Remember, this is starvation mode right now. The person hasn't eaten for a while. The glucose levels in the blood have dropped. So one of the things that we're seeing is we're going to start to break down the triglycerides that we're storing in the adipose tissue. And when we do that, notice the liver is taking those triglycerides. It's using the glycerol to make more glucose. What process was that again? Neo, because it's new genesis. We also see more gluconeogenesis as we take our proteins from the muscle, break them down to amino acids so that we can use those in the liver to make more glucose. So that's also gluconeogenesis. We're also seeing glycogen being converted to glucose here in the liver. What was that process called? Glycogenolysis. Good. We're almost done, guys. Notice that not all of the lipid breakdown, though, is being used for that gluconeogenesis, right? The fatty acids are going to be used to help us make ATP. So for this lipolysis, or fat breakdown, is also helping us with cellular respiration. And as a byproduct of that, it's showing us the ketone bodies building up.
Why does it have two arrows? Like, why does it have two arrows for the ketone bonds?
Because it's showing you that the liver can use those fatty acids to make ATP. It's also showing us that some tissues can use the ketone bodies to make ATP. But if we're in this starvation mode where we have more fat metabolism happening with these high lipolysis levels, then we're going to have the ketone bodies building up. We're going to have something called metabolic acidosis. So regardless of whether it's metabolic or respiratory acidosis, what's your blood pH going to be like?
Between 7.
Lower than 7.
pH lower than what?
7.35.
7.35. Very good. Remember, blood has a normal range.
Between 7.35 and 7.45.
pH of what? 7.35 to 7.45. So if it's below that range, it's acidic, it's acidosis. Also, if I tell you about a specific substance, so this is a good place for one of those thinking questions, right? Because we have two different conditions that have something in common. They have that lower pH. But if I tell you that your patient has, say, a pH of 7.2, and in their blood you also see high levels of carbon dioxide, what's the condition? Respiratory acidosis, exactly. If instead I tell you that there are high levels of ketone bodies, it would be metabolic. And that's kind of a more generic term for the condition. Some of your books are going to specifically call that ketoacidosis, yeah.
So diabetic.
Because there are a bunch of things that can be made in the body that are acidic. If the body makes it and it makes the blood more acidic, that's a metabolic effect. And you were bringing up diabetics. Diabetics usually do have ketoacidosis if their glucose levels are not being regulated properly, right? And that's why, because remember, with diabetes mellitus, that's starving in the land of plenty. The glucose in the blood is fine, but they don't have the insulin working, or not enough insulin to help the body use that glucose. So what does the body do? It starts to use what it can. It uses the fats. So we have more fat metabolism going on, and then we have these ketone bodies building up, which is going to affect blood pH. Don't forget about metabolic and respiratory acidosis, because they're going to come up again later when we study things like urinary system and how we regulate pH. We have studied what can happen to make things go wrong. We haven't studied yet what the body will do to compensate for these things. So later we're going to see what compensation is going to happen to try to maintain that homeostatic range for pH. That's where I was going to leave us for today. I know it's been kind of a long lecture. You guys have definitely earned your break. Take your 10 minutes. I'll see you back in here at 10:30 to go over the last slides for lab. If you do have a question, we can talk about those during the break. Thank you, guys.